So the title of this lesson is called Application of Biotechnology. In other words, what it means is how are we using biotechnology to to our advantage? Okay, to our advantage. So let's start. Okay, now I know most of you do not look at when you buy food. When you buy, let's say, spaghetti sauce, right? Spaghetti sauce. I look at tomatoes. I think spaghetti sauce. You don't look at read the labels, right? You don't read for like calories, maybe some. You don't read for like the ingredients. You don't read things like in specific, like it contains this sugar, that salt, it contains this and that. Most people don't. I remember when I was your age, I never, ever, ever, ever looked at the label. Now, so the question is, how many of you think you have eaten GM food, genetically modified food? Okay? You think you have? Raise your hand if you think you have. Okay. I see about 50%. Now, I'm willing to bet you, we have 26 people. I'm willing to bet you that about 100% of us have eaten it. 100%. 100% of us have eaten it. Because most of the food that are staple food are food that are, is a food item that we eat on a regular basis. Kind of like this. Rice is a staple food. Potatoes, corn, soy products are staple food around the world. Okay, around the world. There's certain people, they eat rice every day. You know what I'm talking about? Yes, you know what I'm talking about. There's certain people who eat corn products every single day. There's certain people who eat certain types of wheat because they eat bread every single day with every single meal. Certain people eat potatoes. Those are staple food. And so a lot of those staple food, about close to 100%, okay, close to 100% have been genetically engineered. That means that what you eat, although it tastes like potatoes, corn, rice, sugars, wheat, bread, things like that, but it is genetically engineered, okay? So we use the term GM. GM means genetically modified meaning that it's been genetically engineered, changed. Most of the time, it's for the better for the organism, for the species, okay? Now, a lot of the things that you and I have also been genetically engineered. Things like our clothes, things like specifically material would be something like cotton. Cotton is a highly genetically modified product. Cotton is in a lot of the items that we wear, right? Cotton. This is cotton. This is, I don't know if this is maybe polyester or something like that. So if you think of all the foods, so what I did was I did some research. I wanted to find out what are the food with the highest level and common food that you and I eat that are genetically modified, GM, okay? So, and what's the purpose behind that? So number one, this is what Google told me. Google said that we have GM food is because we make it better. It is better, right? Food that are genetically modified, they are better. They grow, they last longer, they grow bigger. They're less expensive because they're easier to grow because they resist pests, bugs, chemicals, the sun. They're more nutritious because we add genes to make them more nutritious. So it's all good. It's all good for the environment. Now, what about the statistics? Okay, so I look for only crops and animals. Okay, because that's our main source of food. Now, I found that in 2016, 94% of soybeans are genetically modified. 94%. It is very, very high. Soybean is one of the top. It's a cash crop. Okay, there's a lot of people around the world that eat soybean related products. Okay. And then look at cotton, 89%. My goodness, that's a lot to make your clothing and things around your house. 89% corn. I love corn tortilla. I love it. I love naan. I love burritos. I love tacos. I like that stuff. I like Taco Bell. I know it's not the real deal, but I like chicken quesadilla. Okay. Now, milk is 30%. Okay. And then if you look at fish, 99% salmon. Salmon is 99%, not just salmon. Tilapia, cod, many of the fish that you and I consume are farmed. They are farmed. That means that they're not caught in the wild. These fish they're farmed. It's just like a farm, but it's in at a lake or like a stream or the ocean, and they farm them, and so they feed them with certain chemicals so that they can survive, so they can grow bigger, they can reproduce more eggs, things like that. And then I wanted to look at animals that you and I eat, like chicken, cow, pigs, right? Chicken, cow, pigs. That's our main food, right? Animals. So I wanted to know, what do they eat, those species, if they're not genetically modified? I found they eat 95% of the crops that are GM. So the animals that you and I consume, they eat 95% of the crops that are GM. So does it affect the animals? And then we eat the animals, we might get something? I don't know, okay? I don't know. Now, I have a short list for you. Look at all of these GMOs, okay? Genetically modified organisms or genetically modified materials, right? If you look, I mean, all of these have been genetically modified. All of these. Now, some more than others, of course, but I think most of our food something has happened to it. I think most of our food we buy at the grocery store have been modified. If you ever get a chance, okay, I just learned this, okay, recently, maybe a year ago. My mom has a friend who knows a woman who has a farm up in San Bernardino, okay, up in San Bernardino. And the lady has a chicken. She has hundreds of chicken. And she sells her chicken. And these chicken that she raised, she feeds them with, I'm assuming natural stuff, organic stuff. I don't know. But my mom bought me 2 dozens of this egg from those chicken up in the farm. And then I buy my eggs at Costco, okay? A lot of us buy at Costco, right? At Stater Brothers. I buy them at Stater Brothers too. And when I saw the egg, I put the egg from Costco versus the egg from the farm. The egg from Costco was 30% bigger than the egg from the farm. And I'm like, what the heck? So both chicken, but then when I did a test, I made an omelet, okay? I opened up and I ate the egg. The egg that I got from the farm was sweeter than the egg I got from Stater Brothers. Distinct difference. You can see there's a difference in the egg. Now, did I do a scientific research to find out why? No, I didn't. But I just noticed a difference between the 2 eggs. Now, I've seen it in zucchinis. I've seen it in papayas. My parents have a little farm, in their backyard. They're retired. And they grow a lot of fruits. And I come over and I go shopping there for fruits. And I've eaten their papaya, and they don't use any the chemicals. They use fresh stuff, and their papaya is sweeter than the papaya I buy at the supermarket around here. There's a difference. So, what do I know, right? I'm not a botanist. I'm not a plant scientist. So I don't know, but I do see a difference, okay? Now, but overall, has genetic engineering, has biotechnology, has it helped the society? Has it helped humans? The answer is yes. It helped more than humans. It helps many other species. And if you think of biotechnology, it's just like the technology, the tech stuff like monitors, computers. It is advancing almost at the same rate as computers. Because every year, every 6 months, they discover something. They discover something new. They discover a new disease. And so biotechnology today is so advanced that we are capable of testing almost any disease. We were able to create transgenic animals, which means an animal that has DNA from another species. Okay, kind of like this pig here. This is a transgenic pig. These are transgenic crops. Now, these transgenic crops, rice and wheat, they have cells, they have genes inside their DNA, which prevents them from uh, from from being destroyed by a certain bug or certain bugs that used to eat these crops. They don't eat them anymore. Maybe because of some smell, some gene. I don't know. I eat it. I don't have a problem. I don't smell it, so I don't have a problem. So these crops preventing and treating disease by making food more nutritious. These rice and grains are more nutritious, and creating strains of mosquitoes, genetically modified mosquitoes that's able to carry a certain disease, and then when they mate, they kill their mate, or it prevents them from transmitting the Zika virus, because mosquitoes are notorious for spreading diseases because they inject you, the females. They inject the reason why they inject you because they need food, nutrients from your blood, so they need to suck your blood. The males don't do that stuff, but the females, they have to suck your blood, and so they spread mosquitoes. They spread easily throughout diseases throughout the world, and so by engineering mosquitoes to kill mosquitoes, or to engineer mosquitoes to not be capable of spreading a certain disease, that is a huge improvement. Now, around here, we don't have a big mosquito problem. Yes, we don't have a big mosquito problem. Go and live in Mississippi. Go and live in Alaska in the summer. Go and live in Texas in the summer. Go and live in Louisiana in the summer. Go and live in Florida in the summer. David, if he lives over in Florida, David is fresh meat. He's like filet mignon. Filet mignon. That means the best cut of meat to the mosquitoes. Go. So, how many of you know what I'm talking about? How many of you have lived in another state in the South? Somewhere it's humid. Okay, so you know what I'm talking about. Mosquitoes, they kill me. Okay, they kill me. Okay, so I take my wife. Now I'm from the South. Okay, I grew up in the South, and so I know how mosquitoes are. They fly up to your face, and they don't care. They fly up to your face and said they let you know. Well, you don't understand, but they talk to you. They're like, I'm going to suck your blood, and there's nothing you can do about it. That's their attitude, okay? And so I took my wife to Florida, and she almost died. No, not literally, but she had to run, and then she had to go get the spray, and spray it on so much it affected her skin, okay? That spray that you put on, she had to spray so much. Now, she's a California girl. California girls cannot survive, or even people cannot survive in the South. You know what I mean, right? And the mosquitoes are big, guys. We're not talking about little peewees, okay? Here, we're talking about big ones. They fly up to you and they wave at you before they suck your blood, all right? That is a huge advantage for human. Now, the next thing is, now that we have all this technology and understanding of biotechnology, we can manipulate genes. We can fix a gene that's bad, defective. We can replace a gene that's bad or defective using a technique called CRISPR. I talked about this before. CRISPR, right here, CRISPR. This is an acronym for a type of engineering, genetic engineering, where what you do is you take a person's DNA and you edit it. You either destroy it or you fix it. Okay, that's CRISPR, right? CRISPR. And so that created a branch of genetics called gene therapy. Okay, gene therapy is the study of genes, defective genes, disorder genes, disease genes, and then what you do is you find way to make them, to avoid the disease completely, or to help you fix those disease genes. That's gene therapy. Okay, it costs some money. Okay, it's gonna cost you. It's kind of almost like a cosmetic surgery. Okay, so gene therapy is geared toward people who have who within their family they have a history of diseases, and so you go to a gene therapist, and they analyze your DNA, and they give you pedigrees. They give you they give you a family tree. They do a karyotype. They do all that stuff to let you know the probability of you giving your kids, your son, your daughter, a certain disease. And they can even edit. They can edit your defective genes. before they put it back into the egg, so that they put the egg back into the mother, and so that she's pregnant with the not with the bad genes. The bad gene has been deleted or fixed. Okay, that's gene therapy. Now, so what do they use? What do these gene therapists use? They use a technique called DNA microarray. Right here, DNA microarray. Okay, DNA microarray. This technology started when I was in college. My second year in college. Okay, on my second year in college, I studied this. I read about this, but during that time, they knew nothing about DNA microarray. Nothing. We knew nothing about it, and we were trying to research. And I was maybe about 19, 20 years old, something like that, 19 years old. And I was so lost because the teacher that was teaching it, he didn't even know about it. But today, I mean, it's like 30 years already, almost 30 years, 20 something years. Now we know a lot more. So this is how it works, okay? Now, so humans, we have all this DNA, right? We have like 20,000 genes, right? 20,000 to 20K genes. And we have all these DNA. We have thousands and thousands, billions of these bases, right? And so these genes are found inside your cells, okay? Inside your cell, here's your cell, right? Now, so this is your cell, okay? Now, you have each gene carries 20,000, each cell carries 20,000 genes. Now, so let's say if this is my skin cell, right? If this is my skin cell, does this cell produce all 20,000 genes? The answer is no. It only produces genes that are related to my skin because this is my skin cell, right? Because we have thousands and millions and millions and millions of genes, different types of genes, different types of cells. And each cell, depending on what it is, produces only a certain type of genes. So my skin cell only produces genes that are for my cells, my skin. That's it. Now, so what they did is they do a DNA microarray. Now, only the genes only the genes that produces the protein for my skin are activated. So here is gene number 1. It is for my skin. Let's call it B. It's going to make protein B for my skin. Here's another protein gene. It's going to make protein A for my skin. Here's another one, another gene. It's going to make keratin K for my skin. Now, what about the other 20,000 or 15,000? They are not activated. They are suppressed. So what these gene therapists do is they take the skin cell of the person who has a history of skin cancer. They take all the messenger RNAs. So each of these genes, each of these proteins, each of these genes that are activated in the skin cell are going to make a messenger RNA in the cell. So what these scientists do is they take the messenger RNA out and they put it into a tray like this. They put each of these trays here gets a messenger RNA. So one messenger RNA, here's another messenger RNA, there's a third messenger RNA, there's a fourth messenger RNA. You guys get the idea? So not all of your genes are expressed, only the genes that are responsible for that cell are expressed. So let's say 8 different types of messenger RNA are produced in this cell only because it's my skin. So then what they do is they study each one of these by adding chemicals to it, by analyzing it, by sequencing the sequence of the DNA, and that's DNA microarray. DNA microarray is to study only active genes by looking at their messenger RNA. You study active genes only by looking at their messenger RNA. Messenger RNA will produce a protein. Okay? So, if a person is known to have a certain heart problem, right? Let's say in your family, you have a heart problem. A lot of men in your family get heart disease. So what they do is they take heart cells and they look at active genes of the heart of that family only. They want to look at every single gene. Why is it different from a normal person? So they compare me, my family, who have heart attacks, to, let's say, Raphael, heart genes, and they want to compare. So they compare a person with cancer, heart disease, to a normal person, heart cells. And then they want to see. And then when they compare, after they compare, they find that in my family, there's an extra gene. That gene is, let's say, called H for heart attack. But they don't see that gene in Raphael's family. So that means that this is the problem. This gene is the problem. So what they do is they kill the gene. They kill the gene. They kill that gene in the baby before the baby is born, before it fertilizes with the egg, before the zygote. So they find all the H gene, that heart attack gene, they kill it because that's the gene that causes the disease. Now, what does it look like when they put it into the computer? Now, all this is now automated. It uses machines, computers to do this. DNA microarray, when I was a student, long time ago, was done by hand. It took forever. Now, everything is done by computers. And so it's very fast. So you see each of these dots here, each of these square dots, light dots, each of those dots here, they represent one protein. So one cell can produce a lot of proteins. They put each protein in each of the cell, and they study each one of them to see, and then they compare it to a normal person family. And they want to compare to see, wait, this family has this gene, but that normal family does not have it. No one in the family has cancer. No one in the family has heart disease. No one in the family has diabetes type 2. No one in the family has cystic fibrosis. No one in the family has Down syndrome. They compare it to my family. They look for the gene that are found in my family, and that's how they know. And so they try to use CRISPR to destroy it. Now, another advancement in biotechnology is forensic science. Now, forensic science is the science of the crime scene evidence, okay? So at a crime scene, you find very little DNA. Now, forensic science also branches up into other things, like identifying people, identifying body parts. Let's say you find like a bone, or you find like human tissue, you can ID the person. So forensic science uses a technique called DNA fingerprinting. It started in 1980 by a professor named Alex Jeffries. I think it's Alex Jeffries. He was from the UK, and he used it to solve a case in the 1980s, a rape case. And he was able to convict the guy who raped the woman and killed her. Okay, and so how do you ID somebody? How do you use DNA fingerprinting? So this is how it works. Now, you and I, we have 20,000 genes, right? 20K genes. No, so you and I have 20,000 genes. All humans have about 20,000 genes. Now, if you want to identify a person, you don't compare their genes. So if I take my genes and I compare it to Nu's genes, she and I would be almost identical. We'd be 99.999% identical because I have black hair, she has black hair. I have skin, she has skin. I have 2 eyebrows, she has 2 eyebrows. I have eyes, she has, you see what I mean? I have bone, she has bone. So she and I did the same. So DNA fingerprinting does not look at the genes. Instead, it looks at the other parts of the DNA that is not for genes. Those are called junk DNA. The junk DNA is the region of your DNA where it does not make a gene. All of us have it. So in between each of the genes, there's junk. So it looks something like this. So here's gene number 1, A, gene 2 is B, gene 3 is C. person A. So this is person A, okay? This guy here, handsome guy here, A, and this is person B. He is B, right? So here is his gene A, gene B, gene C, right? In between the genes, people, in between the genes, there's a region. This region is called junk. Junk DNA is a region of DNA that doesn't do anything. It's just junk. That's the region that's different between you and I. You and I are different in that region. Now, if you compare the junk region of, let's say, a brother or sister, you would have the same junk. This person has 12 repeats. Somehow the sequence here repeats 12 times between gene A and B. That's person A. But if you look at person B, this man here, and they're not related to each other, this person has 9 repeats. And it repeats TTAA, 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 TETTAA, TTAA, 9 times. And then there's another repeat, 6 times. AAGG, 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 6 times. But his is different. You guys see what I'm doing? So if I compare my DNA, the gene region, to Jenny's, she and I are the same. If I compare to Nathan, he and I are the same. If you look at it, you would think that he and I are brothers, or we're in the same family. So you don't look at genes. You have to look at the junk DNA, which is the DNA that does not represent a gene. It's the DNA between the genes. And if you're in the same family, you have the same repeats. But if you're in a different family, you have different repeats. By comparing enough of these repeats, you identify the person by using a gel. So you take the DNA of 2 people, different people, you cut it with the same restriction enzyme, and you put it into a gel, you separate. If it's the same family, you should have the same bands, because it's same family, same DNA. But if it's a different family, look, 12 repeats, 9 repeats, 4, 6. It would be different. When you run on the gel, it would be different. It would not be the same. So therefore, that's how you know the 2 people are not related. Now, how many repeats should you compare? The more repeats you compare between 2 people, the higher the probability. That means the more accurate it is. So you don't compare 2 repeats. You compare like 10 repeats. Now, 2 strangers cannot have the exact same 10 repeats. You might have, let's say, out of the 10 repeats, 2 strangers, you might have like maybe 1 or 2 bands that are the same. Like I have 4, Nathan has 4 repeats in a certain area. But that's coincidence. But if he and I were to compare 10 regions, we would not have identical 10 regions of repeats. Nathan, are we good? Guys, does that make sense? That make sense? And that is called DNA fingerprinting. And you know it because you run the